Hello, welcome to Regulatory Affairs 101. I am Nancy. Today, we are walking through the FDA's draft guidance on using minimum residual disease and complete response as endpoints to support accelerated approval in multiple myeloma. This guidance, released in January 2026, reflects FDA's evolving thinking on how deeper response measures can meaningfully accelerate access to effective therapies. This guidance outlines how MRD and CR can serve as primary endpoints in trial, supporting accelerated approval. Importantly, it applies to both drugs and biologics and focuses specifically on MRD negativity in patients who have achieved complete response. And FDA is clear, this is not about patient selection or treatment decisions. It's about regulatory endpoints. Multiple myeloma represents nearly one-fifth of hematologic malignancies in the U.S., and outcomes have improved dramatically with more than 20 new therapies since 2003 and median survival now reaching 7 to 10 years. But with ORRs routinely exceeding 60 to 90%, traditional response-based accelerated approvals are losing discriminative power. MRD offers a deeper, more sensitive measure of disease burden, and MRD negative status consistently correlates with improved PFS and OS. That's why the field is shifting. This guidance doesn't stand alone. It builds on FDA's 2020 MRD guidance for hematologic malignancies, the 2024 draft accelerated approval guidance, the long-standing expedited programs framework, and the 2023 oncology accelerated approval trial considerations. Together, these documents form the regulatory backbone for MRD-driven development. In April 2024, ODAC unanimously agreed that MRD is an acceptable endpoint to support accelerated approval in multiple myeloma. The committee discussed timing of MRD assessments, appropriate trial designs, and disease settings. They also noted that CAR-T data weren't included in the meta-analysis, so applicability to that class remains uncertain. FDA supports MRD-based accelerated approval in both single-arm and randomized trials, but strongly prefers randomized designs because they provide cleaner safety and long-term endpoint interpretation. Trials should be fully enrolled before MRD analysis to avoid operational bias, and sponsors must justify the expected MRD effect size, ideally using meta-analytic data. For combination regimens, contribution of components must be demonstrated, and for multi-phase regimens, the design must allow evaluation of each phase. MRD must be justified for the specific multiple myeloma population. FDA is explicit that there's insufficient evidence to use MRD for accelerated approval in maintenance therapies, smoldering myeloma, MGUS, or extramedullary disease. And if you are proposing a biomarker-selected population, you will need robust natural history and outcome data to support that choice. Trials must enroll patients with measurable disease and pre-specify the MRD assessment schedule. Bone marrow sampling must be adequate to confirm CR, and FDA recommends an independent review committee for CR assessments. MRD negativity should be assessed at CR or within a 3-month window using a sensitivity of at least 1 in 10 to the 5th cells. Imaging-based MRD and substandard MRD negativity are still exploratory. Statistical expectations are clearly defined. Sponsor must pre-specify the MRD threshold, summary measure with a 95% confidence interval, the analysis time point, type 1 error control, and missing data handling. In randomized trial, differences in MRD negativity rates should be evaluated with appropriate statistical tests such as CMH or exact methods. FDA also expects sensitivity analysis, including alternate MRD thresholds, different denominators, and MRD irrespective of CR. Missing MRD data must be counted as missing in the primary analysis and included in the denominator. Imputation is allowed only for exploratory purposes and must be pre-specified. MRD assays, whether NGS or next-generation flow, must be analytically validated. FDA expects data on accuracy, precision, sensitivity, specificity, and DNA input requirements. Analytical sensitivity should be at least 1 log below the MRD threshold, and because sequencing assays can have calibration failures, sponsors must ensure robust sample quality. IDE requirements may apply when investigational MRD assays are used. CR remains a strong prognostic biomarker, and FDA confirms that CR rate can support accelerated approval. CR should be assessed as an overall rate, not at a fixed time point. And durability should be measured from CR to progression or death. Verification of clinical benefit is essential. FDA expects confirmatory trials to be underway before accelerated approval is granted. Sponsor can use 2-trial model, a single-arm MRD or CR trial followed by a randomized PFS or OS trial, or a single randomized trial that supports both accelerated and traditional approval. The single trial model may allow faster verification. It strongly encourages early engagement, especially around MRD assay validation, trial design, endpoint justification, and statistical planning. This is an evolving area, and early dialogue reduces risk later. In summary, MRD and CR are now recognized as acceptable endpoints for accelerated approval in multiple myeloma. Randomized trials remain the gold standard. Assay validation and statistical rigor are essential. Confirmatory evidence must be timely, and early proactive FDA engagement is critical for success. For more detail, you can access the full draft guidance on the FDA's website. Thank you for watching. If you like the content of this video, please like, subscribe, and share. Have a wonderful day.